So can computers predict our evolutionary future? I'm Andy Ellington. I'm with the Center for Systems and Synthetic Biology at the University of Texas at Austin. I think that much of my career has been inspired by trying to do what's called sometimes directed evolution. I try to use evolution as a tool to make things. And in this regard, as others were speaking, and I've really enjoyed the talks, I was thinking that one of my early inspirations, and something that may be a touch point for you as well, is the early George R.R. Martin novella, Sand Kings, which of course shows how you can produce via directed evolution new forms. Now, nature has already shown us this well in advance. Nature is this cacophony of forms that exists, and underlying that cacophony of forms that we see and appreciate and respect are the underlying molecular machineries. So for any one of those organisms on the left, underlying it is a wide variety of protein structures, all of which are quite beautiful in their own right. Now, what's sort of interesting is, as we've gone along and studied the biology, the molecular biology of these organisms, we've literally reached a stopping point in terms of the discovery of new protein structures. For the most part, over the last several years, no new structures, or very few, I should say, new structures have been found. And it seems as though we now have a closure on the sequence and structural information that makes up all living systems. And again, that's probably a bit of an overstatement, but certainly, even though there's been a gross increase in the amount of inquiry, there's been a decline in the actual discovery of structures. So from my point of view, what this means is we now have a full set, a full deck, which we can apply artificial intelligence tools to. And what my lab has done is applied a very simple set of artificial intelligence tools, machine learning tools, known as convolutional neural nets to protein structure. And all convolutional neural nets do is they're these sort of things you see when you feed in a bunch of pictures of cats and you teach it to look at a cat. Well, all we did was take in this structures of proteins, of which there are very many, and then teach the neural net how to look at proteins. And so rather than catitude being the output, it's proteinitude. And in particular, proteins are composed of individual amino acids in individual positions. And so we were able, on a position by position basis, to look at the amino acid-itude of a protein. So we were able to say, for a given position on a given protein, hey, there's a lysine there. Does the artificial intelligence predict that there should be a lysine there? And we got pretty good at this, to where we were repredicting the known proteins with about 80% accuracy. But what about that other 20% of cases? What we were finding was the machine learning tools were predicting substitutions that made better proteins. When we predicted, instead of a lysine, a leucine, those are two different amino acids, and we'd make the substitution to a leucine, the protein would often work better. And that's shown there along the bottom. Now, that's work that we just put out on bioRxiv. It's under review at a journal. But we were able to take a plastic degrading enzyme, and by using an artificial intelligence tool, using this machine learning tool, predict variations, predict substitutions in this protein that greatly increased its thermostability, and eventually allows it to pretty much completely chew up plastic packaging within about 48 hours. So this is a product not of directed evolution, which is what I normally do, but of machine learning applied to protein sequence and structure. Another example of this, and I did want to credit my two students who are really incredible at this, Danny Diaz and Simon Dolsnitz, was to take Google's AlphaFold, which is a remarkable program that can essentially fold up protein structures. And what we did is we took some directed evolution results for another project. This was a project where we're attempting to make non-addictive analgesics, essentially opioids that aren't opioids. And so to do that, we've been working on identifying protein structures that can assist with that. Simon undertook a directed evolution project, which then Danny was able to, because Danny's my machine learning guru, able to show that AlphaFold could correctly recapitulate what we'd done by directed evolution. And not only that, he was then able to dock in alkaloid structures and show that what Simon had found experimentally, Danny was essentially able to predict. So this is my final slide. It's very wordy, but it gets, I think it's going to get the point across. We are now really at an inflection point. I think you've seen that in several of the other talks now, how AI is radically reshaping our world. And what's surprising to me is we can now sort of retrodict portions of phylogeny. We can predict the sequences and structures of ancient proteins. I just showed you we can predict mutations that lead to improved function. We can eat plastics. We can do a variety of things now based upon AI. We can validate directed evolution experiments. What evolution tells us, we can now find out in sort of a retrodictive fashion with AI. We've now moved beyond protein structure to ligand binding. That was the docking of the opioid-like compounds. So machine learning coupled with synthetic biology is an incredibly powerful combination from my point of view. And this leads me to what I just wanted to tell the SciFu audience. Based on what I'm seeing right now, I think it really should be possible to retrodict large portions of phylogeny, to essentially have a computer look backwards into our history, to look at the phylogenetic tree, to look at that great cacophony of organisms from an artificial intelligence point of view. And if we can do that, then we can do the same thing in a forward direction. We can look forward into our own evolution across proteins and eventually across genomes. And I think that this will allow us to do all sorts of interesting things. Even just listening to these talks, the question that I've asked myself for a long time is, if forced to, could I design the genome of an organism that would work at higher temperatures? How would I do that? Of course, we have a lot of organisms that work at higher temperatures, but let's say we need a particular bacteria that needs a particular ground up food supply that operates in the Sahara Desert. How would you make the genome of that organism? I think we are now with AI at the point where we can at least consider that. And I thank you so much for your consideration.